Okay, class. Today we're going to be doing a lecture on Chapter 5, the prokaryotic world. And just as a reminder, the prokaryotic world includes the domains of bacteria and of archaea. So, after reading this chapter, you should be able to identify the major shapes of bacteria, explain the reasons for staining cells, and most importantly, the Gram stain. Identify and explain the function of bacterial surface structures and appendages and their cytoplasmic structures. Describe binary fission and explain why the four phases of bacterial growth curve. To be able to write a short paragraph describing the unique properties of endospores. Okay. Differentiate between growing the bacteria in a broth or on agar surfaces. And talk about the differences between selective and enrichment media, which are all different ways of growing the bacteria. And give some examples of the domains bacteria and the domain archaea. Okay, so this chart's kind of interesting. It shows a couple of different things. One, it shows when the different organisms came into being. The second most important thing is the amount of oxygen that was present. And thirdly, about when the Earth was created, when the universe created the planets. Okay, so if we start with the very earliest, the origins of Earth, 4 point something billion years ago, and then not too long after that, then we start getting the bacteria. Okay, and of course the bacteria and the archaea were among the first things to be alive. Okay, they're the first things that could organize into excuse me, into cells. And then after a while of just having bacteria and archaea, then we finally get some eukaryotic organisms like some yeast. And please note that as the bacteria arose, okay, then we have this increase in the oxygen levels. Okay, and it wasn't until after the sort of age of eukarya, when the eukaryotes were first present, that we finally got animals on here, and then we finally, we get a large increase in oxygen, so we could have the animals, but it basically is all basically down to the bacterial origins that allows us to have all the life that's present in the Earth. Okay. So let's learn a little bit about the function of some of these types of cells. And one of the first things we're going to do is learn the basic forms that the bacteria take. Okay. So there are three common shapes of prokaryotic cells. Those are the bacillus. Okay. And so a bacillus is just something that is longer than it is wide. Okay, and you can see a single bacillus here, okay, with that's longer than it is wide. Okay, and we can have those in chains or other patterns, and we'll talk about that in a minute. Okay. And that's just an example of what it would look like under the microscope. Okay. Our second form of bacteria are the cocci. So the cocci is a ball-shaped cell, and we can have those again in different arrangements, either singular or diplococcus, or two together. We can have them in tetrads, which is 4 together. We can have them in groups, which is staphylococci, or in chains, which is streptococci. And now the last type would be the spiral bacteria. Those could either be single spirals, which would be called a vibrio, or a couple of curves, which is a spirillum, or a bunch of curves, which is a spirochete. Okay, so your 3 types of bacteria: your bacillus, your cocci, and your vibroids. Okay. Now, to be able to see those under the microscope, we need to be able to stain these things. And the basic point of any staining is to provide some contrast between the cells and just the cover slip of the, under the microscope. Okay. So a simple stain simply just means that it's just one color of dye. In this case, they're just dyeing it blue, so methylene blue might work, and you can use anything, but again, you can see that we've sort of increased the contrast because we can see that blue cell on top of the slide. Okay. Now we can also have differential staining techniques, and the gram stain is the most popular of these. Okay. So in a gram stain, you first dye with crystal violet, which is going to turn all your cells purple, and this is whether you're gram positive or gram negative, and we'll talk about those differences in just a minute. We then fix that color in with iodine, so that keeps the stain in the cell. Then the third step is to wash with alcohol, and so gram negatives will turn colorless, and the gram positives will still be the bluish color. And then you counterstain with saffron, and saffron's a red dye, so gram positives remain purple, whereas the gram positives, or sorry, the gram negatives are red like saffron. Okay. Now, the bacterial world is basically split into these 2 categories. You're either gram positive or you're gram negative. And that's because at the end of the gram stain, in a gram positive cell, you'll be that blue color, or in a gram negative system, you'll be that red color. Okay. And so every cell type has to have that cell color. Okay. So as we look at the structures of the bacteria, let's go through a couple of these. Okay, so there's your basic bacteria cell. You'll have a cell wall, which is the hard outer coating of the cell. You can then have a cell membrane, which is the lipid layer that keeps the cell together. It's the functional outside unit of the cell. And, you can have some sugars called the glycocalyx. Some bacteria have flagella. The flagella are used for moving the bacteria around. Okay, they're a whip-like structure that can make the bacteria move. Also on the outside, you have the pili, and the pili really the only thing that the pili does is have the bacteria attached to a surface. Okay, they look like they could be something that could be used for mobility, but they're really not. It's really just flagella that do the mobility. Okay. Now, on the inside part of the cell, okay, basically the fluid-filled part of the cell is the cytoplasm. Yeah. And then, as opposed to a cell that would have a true nucleus, the bacterial cell have a nucleoid, and the nucleoid is just a concentration of DNA. Okay, it's not a membrane-bound structure. That again is why these things are not eukaryotes. Okay. We can have small pieces of DNA, and those are generally circles, and those circles are called plasmids. And then where we make different proteins for the cell are on the ribosomes. So those are the basic structures that you'll see on the outside and on the inside of a prokaryotic cell. Now, that cell wall, okay, or cell envelope, is one of the most important things about these bacterial types, and that actually is why the cells get divided into the gram-positive and gram-negative status. Now here illustrated is a gram-positive cell wall. Okay, so we have one part, the cell membrane, which is a phospholipid, so lipid tails with phosphate groups. Then these layers here are the peptidoglycan shell, and what you'll notice in a gram-positive, this is a very thick layer in a gram-positive. Okay, and that's why it takes up that stain, the crystal violet, because the peptidoglycan is going to be able to stain with that blue stain. Okay. The other thing you see here, you have some sugar groups, okay, that can either attach to the peptidoglycan or actually attach to the cell membrane. Now, in a gram-negative cell, okay, you actually have two membranes. You have a cell membrane, same as for the gram-positive. Then you have a thin layer of the peptidoglycan. Then there's an outer membrane, okay, and this two-layer system is what's going to go red on the gram stain, and most importantly, it's a very, very thin cell wall here. Okay, now one of the things illustrated here is that we have some proteins in the outer membrane which will allow things to go through to get in to feed the cell. So it's sort of selective, the outer membrane, and only taking up the things that the cell needs to eat. Okay. So again, the cell wall. Where this first layer, or the only layer of the peptidoglycan is called the periplasmic space. And a lot of processes go on in that periplasmic space. These again are sugars on this outside place, the lipopolysaccharides, and those lipopolysaccharides in large part become an antigen in a human, meaning there's something that elicits an immune response. So when you would get some of the bacteria in your system, your immune system will try to fight it off, and it's mostly this LPS layer that is what's reacting with your immune system. Okay. And I've already explained the phospholipid bilayer. Now, that cellular envelope is what mostly controls the import and export of things into the cell. So whatever the cell needs to eat, or whatever product the cell makes, will pass through that cell membrane. Okay. The glycocalyx, this is something called a slime layer. And there's some bacteria who have very major slime layers. Some just have kind of normal, but you can see it when you grow these on a plate. You can see a lot of the slime of that glycocalyx. One of the major things that a glycocalyx will do is to help the cell survive any environmental stresses. So whether it's drying or too much heating, the glycocalyx can help protect the cell. Okay. Glycocalyx can also be used to attach to the surface. Okay. Things that help move the cell around are the flagella. Okay, that's the mobility, and then just to get things to stick to a surface, you have the attachment of the pili. Okay, so the pili are the short ones, the flagella are the long ones that go for movement. Okay, so here's just a summary of the things on the outside of the cell. Okay, the cytoplasm is basically the guts of the bacteria. So this is a combination of a lot of water and any dissolved material that the cell has. Now within the within the cytoplasm, you'll have a nucleoid, and so the nucleoid is your singular circular DNA chromosomes. Okay, and there's no way the cell could survive if you didn't have some DNA inside the cell. Now the second part of the cytoplasm are the ribosomes, and the ribosomes are basically a small machine which are used to make your protein. And there are literally thousands of these per cell. Usually you get a few passed down because a big kind of theoretical question would be, well, where did the first ribosome come from? So a ribosome is made up of proteins, and if you need a protein to make another protein, where did the first one come from? Okay. We can also have things like plasmids, which are just circular pieces of DNA, and these tend to be things that are sort of extra to the chromosomes. Okay, so very often those could be something like an antibiotic resistance or something like that that the cell would need in certain conditions to be able to survive. Okay, and there's a summary of your cytoplasmic structures. Okay, now we'll talk a little bit about reproduction. And so we'll talk about binary fission, which is a major way that prokaryotes divide, and then also endospore formation, which is a tool for survival. Okay. So binary fission basically just means taking two things and making them, or one thing and dividing it in two. Okay. So you start with a single cell. Okay. And then the cell is going to divide its chromosomes. And then make itself into two cells. Okay, so again, the process of going from one cell to two cells is binary fission. Now, when you plot that out in a growth curve, you can see first in this figure, this shows you the number of cells. Okay, so when you start off with this one cell, you then would get two cells, and you just increase, by two every time. Okay, and if you plotted this on a normal piece of log paper, or sorry, of graph paper, you would get this really steep curve. However, if you plot it on log paper, you get a straight line. Okay, so like we said before, the reproduction is in 4 stages. Okay, so you have the very first part is the lag, and so in the lag period there is very minimal increase in the number of cells, but on this graph it looks like there's basically nothing. Then you have the log phase, which is a straight line on logarithmic paper. Then you finally have a stationary phase, which is the point where cell number plateaus. And then you have the decline phase, where the cells start to die. Okay, and not every bacteria dies to the same extent. Not every lag phase or log phase is the same amount of time. Not every stationary phase is the same amount of time. In a way, it would be nice and a lot easier to study the bacteria if all these phases were equal, always the same, but in fact they are always really different, which sometimes is a way of helping to characterize the cells, because they will have very different growth rates, for example. Okay. Now, if you want to be able to survive in a harsh environment, you can form endospores. So here we have, in the red there's a typical bacteria, and then around it are the endospores. Okay, and the endospores are again a heat-resistant, um, drying-resistant form that will help the cells to survive for a very long time. Okay, these are generally only produced by some of the gram-positive species. Okay, so yeah. Okay. And you can see there, compared to the mother cell, those free endospores are quite a bit smaller than the mother cell is. Okay. Now, as we grow those mother cells, we have to worry about what kind of media that we're going to grow these bacteria on. Okay, so we can either do these in broth media, where it's just a liquid. Okay, so there's a test tube with some liquid in it. There are some of the cells are growing there in the bottom. There's a little bit of cloudiness in that liquid, so there are cells in there, but the kind of majority of them are down here at the bottom. They've settled. Okay. Now we can also grow on an agar plate. Here someone has touched a plate, and you can see that there are many bacteria that have been able to grow up. Okay. Now, selective media is a kind of media that allows certain kinds of cells to grow and prevents other kinds of cells from growing. Okay. We have a number of different selective medias. One kind of those are able to select just for the gram-negative bacteria. Okay, so it would be a good example that only gram-negative cells could grow on some of these specialized medias. And I'm not asking you to name those specialized medias, just know that selective media is something that allows only certain types of cells to grow. Okay. So you're gonna the way to make a selective media is to add something that will inhibit the growth of some other type of bacteria. In this case, if we want to grow the gram-negatives, we're going to add something that inhibits the gram-positives from growing. Okay. Okay. So here is a plate that was trying to look for gram-negatives again, and on panels A, B, C, and D, they were all streaked, okay, but you'll see that B and D don't have any growth, whereas A and C do have growth. So those would be the gram-negative bacteria because they're allowed to grow on this type of media. Now, enrichment media is slightly different in that enrichment media allows growth of one type of bacteria more than another type of bacteria. Okay, a sort of basic principle is that nothing grows with all of the known nutrients. Okay, and then you kind of selectively add new nutrients until you can get a cell to be able to appear. Okay, and sometimes you can get lucky and find this one time. You know, can be quite easy. A great example of this was for Legionnaire's disease. Okay, so at first we didn't know what Legionnaire's disease was. We knew it was a type of pneumonia that people were getting, but we couldn't find the bacteria that was growing it. And so with this enrichment culture, one of the things that they did was they slowly added different nutrients, and when they added aluminum, they finally got the Legionnaire disease to be able to grow, and then they had a enrichment media so that those type of bacteria could grow, and they could actually test it and try to find a way to cure the disease. Yeah. And so this would just be an example, something like the Legionnaires, again adding the aluminum, because that's, to have very high levels of aluminum would be fairly uncommon. Okay. So here are some examples of some of the bacteria and archaea. So first, remember our domain structure. There are 3 domains: the bacteria, the archaea, and the eukarya, which aren't shown on this graph. But the bacteria are the bacteria that are sort of more common things. Okay, they're the things we interact with on a daily basis, things that are in your gut. These can be anything from something that causes pimples to something that makes yogurt for you. So again, the bacteria are sort of the common things, whereas the archaea are things that are fairly rare and really unique. Okay, they have some unique property to them. One of the archaea that you actually sort of encounter a lot is the bacteria in the Great Salt Lake. Okay, so in Utah, in the Great Salt Lake, there's a type of archaea called Halobacterium, and Halobacterium only grows with a whole lot of salt around in the media. Now, as long as we've known bacteria for a really long time, and we think they're somewhere between 100,000 and 10 million different species, but no one knows yet. Okay, I mean, that's one of the reasons why microbiology is so exciting, because if any of you would go into a microbiology lab, there is a chance that you could discover a brand new species if you looked hard enough. Okay, so we don't know an absolute number yet, whereas we pretty much know the number of animal species that are present. Okay. Like I said, we only sort of know about 12,000 of those species as of today, but there could be up to 10 million different species out there. Yes. So those are just some of the different cell shapes among the bacteria. Okay, so what will we have learned from this lesson? The shape and arrangement of cell types, and guarantee you cell shapes and cell arrangements will be on a quiz. What kind of staining we may use so we can see the cells better. The structure of different parts of the cell. How do cells divide and reproduce, and also the parts of the growth curve. And why endospores are so good at helping cells survive, and how we can use different growth media to manipulate the cells, and some examples of the bacteria that we can find out there. And that should be a pretty self-answering questions, but are there more different types of bacteria than just little rods and spheres?